This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 781, recorded on July 16th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello there, Vincent. Good to be back.
Yeah, welcome back. We missed you.
Thank you. Well, the feeling was mutual, actually. I'm looking out the window. It looks nice out there, but don't you dare step outside unless you have a portable air conditioner with you. It's really hot and humid. It reminds me a lot of where I grew up in New Orleans. 33
Celsius.
It's incredible.
I just walked to the mouse house and wow, it's really hot.
Yeah.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. Great to see you back, Dixon.
Thank you.
The weather here is the same. I walked down to town to get some lunch earlier, and I think I'm still cooling off. My office is usually very chilly, so I'm pretty happy in here right now after having been outside, but maybe by the end of the episode, I'll have to wear a sweater. So we'll see.
It's amazing.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here, and also welcome back, Dixon. Great to see you.
Good to see you.
And it is wet blanket weather here too. It's 90 Fahrenheit, 32 C. The dew point is 70 Fahrenheit.
Oh my God!
So that's humid.
If you think this is hot, just go out west.
We're also having intermittent thunder showers. One just rolled through a few minutes ago.
Right. This is a cold wave compared to what they've got.
Oh yeah. Nothing's catching fire yet.
No, but
not around here.
Bite your tongue.
Yeah.
We don't have blister bark beetles either, so we don't have a lot of dead trees to catch on fire. So that's good.
Dixon, there were some episodes where we needed you. We were talking about mosquitoes on one and various animals on another.
I hadn't caught up. I'm sorry that I haven't caught up.
No worries.
It's all right.
I spent a week and a half in a place called Kessler Rehab Center, and it's got 3 offices, and one of them is in Saddlebrook, New Jersey, off of Route 80. I can't tell you how exciting that was to actually find out what an occupational therapist does, as well as a physical therapist. I knew with physical therapy, the occupational therapy comes to my room and she says, "I'm your occupational therapist." I says, "I'm retired." She says, "You know how many times I've heard that?" So then she proceeds to teach me how to carry out my daily life, and that's your occupation. And I guess a lot of people know that, but of course, never having been a patient in a hospital, it was a brand new experience for me. So
I could have used that 50 years ago, how to carry out my daily life.
How to cater, yes. I think most people in their 20s could benefit from occupational therapy.
I think I could certainly get some benefit from it.
Whatever they showed me is nothing compared to what I have to learn at home.
I could still use some occupational therapy occasionally.
It's all right. My wife is a great OT person.
Well, Dixon, I'm glad you're back, and we'll get you onto TWiP. We have an episode coming up. You know, we did it. Daniel and I did a TWiP, just the two of us.
I felt badly. For missing it.
You felt badly because I don't know anything, and it was just Daniel.
You do so.
But we're going to have one next month, and we'll have you back. That's great.
Was that the Plasmodium vaccine?
Yeah, Plasmodium mRNA vaccine.
mRNA.
How about that? And it's going to be everywhere now.
It's absolutely great.
Right?
It's absolutely great.
And it does not come with microchips, folks.
No,
it doesn't. No. We don't know where you are. And frankly,
by the way,
we don't care where you are.
You can get microchips that you embed.
Oh, really?
The cat, both our cats have them.
Yeah, our dogs have them too.
I wish my car had one.
No, they're commonly used in pets, so that if it gets lost and the collar falls off, they'll be able to scan it and find it. But that's just an RFID chip. It doesn't actually.
The one I loved the most was they had an implantable chip for cattle. Measured their temperature to see if they had shipping disease, which is brucella.
That's right.
And they actually got it.
Well, if you really want to track people, folks.
That was exactly what I was going to say.
Exactly.
I mean, everyone, you're carrying it all the time, right?
You got it. Just go on Twitter. You can find yourself anywhere you want.
By the way, before we start our papers, I just wanted to mention, you guys hear that in Tennessee, they're not going to push vaccination anymore? It came from an official.
Absolutely crazy.
I mean, that should be punishable in some way because denying people public health is horrible, right?
Yeah.
Science has got a bad rep, and it should never have a bad rep, but that's what's going on right now, I'm afraid.
My gosh, that's horrible. And so it's not just COVID vaccines, it's flu and HPV.
It's everything.
Yeah, their public health department is not allowed to promote vaccination of kids anymore.
That's crazy.
Basically, is the upshot of it because it's politically.
I know
that's the place they've got.
But Alan, how does that play out in terms of a national program now?
Oh, well, the CDC is still going to do CDC stuff, and all the public health, the federal public health agencies will do their thing. But what a lot of people don't realize is that the federal agencies don't do much at the state level without direct involvement of the state departments. So the State Department of Public Health, when the CDC pushes something out, they push it out to all the state departments of public health to have them, actually do it. Of course. And so it's just not going to be promoted. And it's not that they're denying people vaccines. They're just not going to promote them because that has been deemed just politically incorrect, apparently.
Of course, as Lori Garrett said, that they don't have to do what CDC says anyway.
No,
so.
Right.
Sorry.
Yeah, well, it just makes me wonder, have they been making changes in their healthcare budgeting for the increases in numbers of vaccine preventable diseases?
Of course not.
It does raise a lot of questions about federal funding for public health programs, however. They could use that as a lever to get them to come back online.
That would get really toxic really fast.
Well, it's toxic already.
No, it would get a whole lot worse.
Well,
if you start strong arming public health on states, then they're just going to become more and more entrenched.
I think a lot of people need encouragement to get vaccinated, right? So it's unfortunate that a lot of people won't. And this is, if you have long COVID, it's devastating. And
no kidding.
This is going to be a huge healthcare burden.
You got it.
And it already is in areas, exactly the same areas that are
Missouri. Missouri has a similar problem.
Missouri and Arkansas. It's really focused in specific areas.
That's right.
And so it's Missouri and Arkansas throughout the South, just areas that, especially rural areas, where people...
My enlightened state of Louisiana, of course.
Right, where people are just not on board with the science of it. And because they've been told by trusted authorities that, this is the truth that they believe.
The Surgeon General actually came on NPR last night and started to talk about misinformation as a crime.
He did. He's sort of been making the rounds, having that conversation. And I think that the point that Alan just made that's so important is that so many people really need to hear information from sort of trusted sources of advice. And I can imagine that the state public health department in some cases might be more trusted and might be thought of as being kind of more tailored to you than a bigger organization. And so it is a real problem that state public health organizations are not promoting vaccines.
As a general phenomenon in public health, the more local you get, the higher your credibility.
Right.
If you go to a vaccine clinic and you see somebody who you meet at the supermarket all the time giving the shots, that's a very, very high level of credibility. If it's somebody from out of town who you don't know and you had to drive to some other location for it, that's a very low level of credibility. And so the federal government coming in saying, this is what we recommend, but the state, as you say, Brian, the state level and down to the local level, much higher credibility. And so when you hamstring those local and state public departments of public health, that's a big issue.
I think we should look up Elvis's vaccination record as a child.
Yes.
And then put it up and say, look, if it was good enough for the king, it's good enough for you.
He's a poster child for polio vaccine, right?
Yes.
Yeah.
Celebrities. Right. I don't know. Have any celebrities
how about Dolly Parton?
Forward for COVID vaccine?
That area?
Well, Dolly Parton.
She's been... Yeah, go ahead, Brianne.
Yeah. I was saying Dolly Parton was involved in funding for one of the vaccines.
That's right.
She is very pro-vaccine, and that has definitely been helpful, but not as much help as we need.
Yeah.
Wow.
All right. Wow. We have a couple of papers today. We have first a nano snippet, something new.
Because we're actually trying to do it in an actual snippet, yes.
This is a paper published in Viruses, a journal that we don't use very much, but has a lot of virology in it.
Is this a single base pair snippet?
Yes.
And it's a open access journal. Actually had the editor-in-chief of Viruses on years ago, Eric Fried. Oh, that was so long ago, to talk about open access and publication. Anyway, this is a paper out of Italy. Do dogs and cats passively carry SARS-CoV-2 on hair and pads?
And the answer is no.
They don't.
Moving to our next paper.
The funny thing is that they go through all the methods and then they have one line of results.
Yes.
Which we'll get to. But I want to say some of the names. The first author is Stefania Lauzzi, and the last author is Saverio Paltrineri. They're from University of Milan and Istituto Zooprofilattico Sperimentale della Lombardia e dell'Emilia Romagna, University of Bari. There you go, the three of them. Now I got my chance to practice some Italian. So the idea here is, how much touch is involved in transmission of SARS-CoV-2? We don't actually know, but remember hygiene theater way back when in the beginning we thought it might. And we know that from a variety of studies that infectivity can last on surfaces for a while. So, some pets rarely get, in fact, cats and dogs, right, get infected quite rarely. But they say here during the first months of the pandemic, Italian websites asked pet owners got questions from pet owners. Could their dogs or cats get contaminated? And in fact, I got a question from a professor here at Columbia. If we walk our dog outside in the city, are they going to pick up a virus on their foot pads? And so that's what this is all about, hair and pets. And, so it's not so much the pet being infected, but the owners, contaminated, because people like to hug their pets and so forth and so on. So
and you can also imagine, maybe there's this worry that if your pet has it, it could contaminate you. So it's not just your owner contaminating the pet, it's the pet being a source of infection for the owner.
The pet as a fomite, yes.
Yes.
So this involves 48 dogs and 15 cats from Italy, COVID positive owners, SARS-CoV-2 positive owners. This is from April to May and October, November 2020. So these
cats were a lot less likely to show up.
Yeah, apparently. So these are diagnosed by PCR and symptoms. And the dogs and the cats could not have any clinical signs. And they could not have any RNA in nasal, oropharyngeal, and rectal swabs, right? So this would be not the pet contaminating itself.
That must have been fun collecting.
Wow. So it was all collected during routine veterinary visits with consent. Yeah, I mean, they got a lot of samples. And they got cutaneous samples by brushing on the skin and hair, from the neck all the way back. And then they brushed the foot pads and the in between, which I can imagine some pets did not like, right?
No.
I mean, when our dogs, if you touch their feet, they pull them right. They don't like you to touch their feet, right?
That's right.
It's probably important for them to keep their feet intact. So,
exactly.
All right. So they take these swabs and they do RT-PCR for the E gene. So it's PCR based, no infectivity, of course. And they have a sensitivity of 11 to 15 copies per reaction with a 99% detection probability using human samples. They did controls to rule out contamination and so forth. And so they have, this is important actually, the time from the positive testing of the owners to the collection of swabs from the animals ranged from 1 to 72 days in dogs and 5 to 68 days in cats. So some of them they picked up early, some of them they picked up later. And then there's a one-line result.
The result.
All cutaneous and interdigital swabs tested negative for SARS-CoV-2 RNA.
Bingo.
All right, so this is a small sample. And they do point out that in a previous study, there was one positive out of 56 dogs. And that was within 21 days of the owner diagnosis. But they say here we had some that were within 21 days. So I mean, I think if you looked at enough, you'd find probably a few, but it's just not very common.
It's extremely rare.
Yeah.
And we don't know how much is there and whether it's infectious and whether it'd be enough to infect you anyway. So
Did they just step in some RNA, or is this the virus? Yeah.
Did the cat step in RNA? RNA is everywhere, of course.
Yes.
Exactly. Cats don't do that, but dogs do, I think.
Yeah.
Anyway,
Cats bury that RNA.
Yes.
Yeah,
and
dogs.
I expect if you screen for more than just SARS-CoV-2, you can probably find something on the footpads of these animals.
sure.
Let me tell you, a lot of parasitic disease entities,
Sure.
protozoans and eggs.
Yeah.
That's a great way to transmit them.
Yep.
Yeah, I was going to ask what other viruses we might think would be transmitted this way or would be found here.
Anything they pick up from the ground, right?
Yeah. Any fecal
guess.
animal virus,
I mean,
I think would
dogs love to smell poop, right?
Yeah.
They do.
You know,
Sometimes they even eat it.
our lawn is full of deer poop, and they will eat it for God's sakes.
That's right.
I
That's
have
true.
to pull them away from it. They will bloody eat it. I don't get this.
Well,
But anyway-
half of the poop of deer is an edible plant. It's only 50% microbe.
It's true. It's true. But so you could get viruses from the deer, I suppose. And there's bird poop. There's mouse, rodent poop. Hey,
You
we
name it.
have foxes running around. We have
That's more
raccoons,
dangerous.
rabbits, groundhogs, all kinds.
Yeah.
Bats. So they can have a lot of things. In fact,
They can.
they should have done deep sequencing on these.
Yeah,
swabs.
that's a different paper. That's
I imagine they would love to survey wildcats too, but it all depends on what it ate last. Or
Feral cats. So are there feral cats around here, Dixon?
oh my God,
They're just
yes.
wild. They're normally normal
Absolutely
cats,
right.
but they're just wild. They have no owners, right?
That's right. That is
Yes.
correct. That
No,
is
that's a
right.
house cat. So the kind of cat like is behind Alan. It's a house cat, right?
yes.
That's right. That's
So
right.
wild cats are like mountain lions and tigers.
Right.
And all.
Yeah.
But we don't have any of them around here, right?
Well,
You probably have bobcats.
I think we have something.
Yeah, you probably have bobcats. They're pretty common. But we
Brienne,
don't.
Brienne makes a scientific statement. I think we have something.
We have something, yeah. No, in this
Out west, they have a lot.
in this area at least, we actually have very, very few stray cats. They're very rare, mainly because we have coyotes.
That's right. Coyotes.
And we also have fishers. We've seen those,
Oh, is that right?
which will also eat cats
That's true.
and pretty much anything else.
Yeah.
So the
Yes,
question is, Alan, if it's a feral cat, you're not allowed to trap it, right? Because it belongs to the U.S. government, right?
No. If it's a feral cat, right. So different rules apply to feral domestic animals, I think.
Yeah.
So those could be trapped. Also, there's no hunting regulation on them. So, I think but yeah, feral cats wandering around could be trapped. And in fact, the local animal shelters do that. They've got a program where they go out and trap any feral. I think in the city of Springfield, like right downtown where there aren't
Yes.
so many coyotes, they do actually have some feral
Not
cats.
many coyotes.
All right, so according
How would
to
you know
the,
they hide so well?
They
According to
do,
the-
but not well in the city.
According to the
That's
New
true.
Jersey Department of Health, there are about 70 million feral cats in the U.S. These are
Yeah.
like your cat
70
behind you.
Oh yeah, no,
Million.
there are a bunch around here that
They're huge.
we see
And
walking around
that's
from time to
very
time.
important for
They-
the wild bird population.
Sure. Yes. The
Eat
other
them up.
thing, the
Oh,
other thing that
look
keeps
at
them
this.
down somewhat in the Northeast is the winters.
Yes.
A lot of them don't make it through the winters there, which is why there's a larger feral cat population in the South.
Yeah.
Wow,
Well,
we
I
have
wonder-
we have bobcats in New Jersey. Look at that.
Yeah, of course
you do.
Of course. So one good piece of news for restoration of natural systems is the fact that we now have a native population of jaguars
Oh,
that crept across the border before the wall went up.
well, the wall is not stopping anything, Nixon.
No, that would never stop a jaguar anyway.
No.
But they would eat the people that were installing them probably. But we now have a native population of jaguars down in the Southwest, which we had, and then they hunted them out of extinction.
Right. And
So
now they're back.
And now they're back.
The bobcat's native to New Jersey. It went down in the 1800s, and now it's on the rebound.
Yeah.
Cool.
Of course, we have
mountain
bears.
lions also.
We have
the
bears.
Catskills.
We have coyotes as well. So they could get a lot of
Exactly
different
right.
viruses.
Exactly.
Yeah.
We have 6 whales that are native to the coast of New Jersey,
Oh,
apparently.
that's true. That's true.
And my wife showed me on Facebook there was a humpback sighted off New Jersey coast. This
Oh,
week.
right.
Isn't that
Vincent,
cool?
don't you remember we had one that swam up to the George Washington Bridge one year?
Did you see it out the window?
There was a humpback whale that actually swam up to the GW bird. But they didn't pay the toll, so they were turned back.
Yes.
Today is
the whales were quite common off the coast of North America into the 1700s, and early whaling did not involve long sea voyages.
That's
right. That was what I thought.
Right.
They would get in small boats, like the small boats they later used from the ships, and row out. And the first type of whales that was harvested off the coast of Massachusetts was right whales.
Exactly.
And they are called right whales because they were the right whales to go after, because when you harpoon them, they floated, and not all whales do that. Some whales, when you harpoon them, they sink, and that's a very bad thing. If you're 6 guys in a small boat, you can't really pull it up from the bottom.
And so a Nantucket sleigh ride, you harpoon it and it goes
that's
for a
when
run.
You harpoon it,
hang
and
on for dear life.
That's larger whales. Usually they take off, and then everybody hangs on to the boat and goes zipping
and
across
then
the waves.
Sound. That's right.
If they sound, they could pull the boat under, right?
They could if you don't pay out slack fast enough.
That's right.
And of
or
course,
if it gets wrapped around your foot.
Nantucket.
This, by the way, is not a digression. It's foreshadowing.
It's foreshadowing.
Exactly.
Nantucket sleigh ride is
you could call it loomings.
Is a great song. Is a great song by Mountain. Nantucket sleigh ride. I love it. You remember that? Anyone remember? You're all too young to remember.
We are all too young.
Yeah. Was that from the original whaling days when you were a kid?
Vincent actually studied with a little oil lamp he had in his bedroom. That's how he got so smart.
So Mountain was a rock band. Have you heard of Mountain?
I have heard of Mountain.
Yes, I have heard of Mountain.
So their big hit was Nantucket Sleigh Ride. Anyway.
Oh, really?
The guitarist was Leslie West, and the drummer was Felix Pappalardi. All right. So I do a live stream on Wednesdays, and there's a guy who I enlisted to moderate. He lives here in New York City. His last name is Pappalardi. So I said, "Are you by any chance?" He said, "Yeah, it was my father's cousin."
Oh, wow!
Isn't that great?
Okay.
Isn't that great?
Wow.
I love it.
Very good.
I love that song. So I actually saw them live in concert in Asbury Park Convention Hall years ago.
Terrific.
All right. So the last thing from this paper, they say our results suggest that pets do not passively carry the virus or play a role in the transmission. And we hope this will avoid unnecessary discrimination against animals, because, people can be kind of not nice.
There was concern early in the pandemic that people would be getting rid of their pets. In fact, what happened was the reverse. The shelters just got cleaned out. They've got no animals left because everybody went to adopt them.
And people began to steal dogs and selling them too, because people really wanted them.
Because there was such a demand.
That was crazy.
Yeah.
Okay, moving on to humans. This is a paper in the Journal of Clinical Investigation, which I thought was kind of neat. Mm-hmm. Efficient maternal to neonatal transfer of antibodies against SARS-CoV-2 and BNT162b2 mRNA COVID vaccine. This is from a big study out of Israel involving the Weizmann Institute, Tel Aviv Medical Center, et cetera, many, Technion, Tel Aviv University, and the first 2 authors, Ofer Baharier and Romina Plitman-Maya. The last author is Michal Kovo. And so many people, have said, is it okay to immunize pregnant women? And, they were left out of the main initial vaccine trials, although now there's actually a trial in Israel that's been done. And many of them were inadvertently included because people got pregnant during the phase 1, 2, or 3, whatever it was. So we do have some data. But it's important to protect them. It says here in the introduction, 80,000 pregnant women have acquired COVID in the U.S. alone. And some data suggests that they're at increased risk for serious adverse effects and even pregnancy complications, right? So now I believe the Israel Ministry of Health is advising that pregnant women should be vaccinated. I think the CDC is as well.
CDC has been recommending that for a while. Yeah, and that's based on the advice of the all the big medical organization, the American College of Obstetrics and Gynecology, concluded that this was definitely a good idea and all that. And we now have collectively worldwide, I'm sure we have the experience of millions of pregnant people being vaccinated. And it seems to be very well tolerated. And we knew going into it that getting COVID-19 was a really bad thing in pregnancy, because pregnancy is accompanied by these immunological changes that we've talked about on past TWiVs, and there's a lot going on. And of course, there's, the fetus could get infected and this and that. So the vaccine was clearly lower risk, and it turns out quite beneficial. And this paper underscores that.
This is about the fetus and neonates and infants who can't be vaccinated, right? But they can get antibodies from their mom. They can get IgG. So, and in fact, Alan, do you remember we did the TWiV in Nova Scotia, and one of our guests talked about immunizing pregnant mothers against RS virus to protect the newborn, because it's so hard to immunize them, shortly after birth. And so here they said, does the fetus acquire antibodies? Which I guess, Brienne, only IgG can cross the placenta. Is that correct?
Only IgG can cross the placenta. There's a specific receptor that actually transcytoses that IgG across the placenta. So it is the only one that will make it.
Is it a FC receptor?
It is. It's called FcRn.
Okay. Appropriately, a nurse receptor. Yeah.
Nurse receptor.
It's the FC receptor for neonatal transfer, is I think how I understand it anyway.
Yeah, I know. I was joking.
Yeah.
Nurses.
But IgA can also go through breast milk.
Sure. I guess
IgA, any others go through breast milk as well?
Really, it's all IgA.
Okay.
Right.
So this is is this true for other mammals as well, Brienne?
Absolutely.
The person you would need to ask that question is Steph.
Yes, I was just going to.
But I think what I have heard from Steph is yes.
How about kangaroos? I don't know.
I have no idea
because they don't have a placenta, right?
That's true.
They're marsupials.
That's true. I have never thought about what happens with antibody transfer in marsupials until this moment.
It goes through their milk. It must go through
the milk, because they crawl out and then they drink milk, right?
That's right.
Yeah.
Okay. Interesting. Okay, so in this study, 1,094 participants from 8 hospitals across Israel, and they collect samples following childbirth, and they have matched maternal and cord blood, right? You can easily put a needle in the cords, pull out whatever you want, and then the mom is there too. Of course, they have to consent to it, but that's what they did, and they could match. And they have an interesting patient set. They have 105 who have been vaccinated and 94 who were not, but they were previously positive and presumably infected.
Right.
So you can compare transfer of antibodies in both scenarios, vaccination versus natural infection, right? So this is a dyad. I love that word. They're dyads, right? The
dyads,
mother-child dyads. And I guess it could be a triad too, right?
That's true.
So they did ELISA-type assays here. They looked for antibodies to spike 1 and 2 subunits, the RBD, receptor binding domain, and then the N protein of SARS-CoV-2. And they look at these data, and then they figure out a percent transfer ratio. They look at the level in the mom and the newborn, and they say, what's the percent transfer ratio? And interestingly, the transmission rates of IgG, S1-2, RBD, and N antigen antibodies were higher in moms who had infection as opposed to, first, who were PCR positive at week 30 compared with after week 30. All right, so higher in participants who were PCR positive in gestational week before 30 as opposed to after. Okay.
And that kind of makes some sense. You would assume that it takes a little while after infection for you to start making an IgG response. IgG is not the first isotype that is generated. And so those women who were infected a bit earlier in their pregnancy would have had more time to actually get around to making IgG in that immune response.
So the total gestational time is 8 months times 4 is 32, right? Wait, 8 times 4 is 32
No, I think
weeks.
it's 40 weeks.
40 weeks, 9 months,
40
9
weeks.
times 4 is 30. 40
Yeah.
weeks, okay.
I think it's 40 weeks.
So 30 is getting towards the end there also. All right, and also when they look at the transfer values, they were better when infections were before 30 weeks, right? So transfer of IgG to the fetus. So that's looking at timing. And then they look at the transfer values. That's infection. That's infection so far. And by the way, what's interesting is when they looked at these ELISA results, they actually found some moms who were infected but didn't know it actually, because they found antibodies to in the vaccine group. They found antibodies to non-spike proteins, and they also found 4 neonates with an IgM response to all the antigens.
Interesting.
Which they say could be either a compromised placental barrier, fetal exposure, or vertical viral transmission. I guess all of those would work.
That makes better sense.
Right.
Yeah.
Well, it sort of depends. I mean, they talk about one of those 4 cases being a preterm birth with rupture of fetal membranes. And so that one, clearly you can imagine how there could be, a compromised placental barrier that was in fact specifically the problem. But I did like that they kind of specifically addressed this, because I was sort of trying to figure out exactly what we were trying to say was happening with these IgM responses in terms of whether these fetuses were infected or not.
Right.
So then they compared this to the vaccination, 2-dose mRNA vaccine, which is 3 weeks apart. And they see a gradual rise in, so they're looking in serum of his mother's, gradual rise in IgG during the first 45 days after infection. And with the vaccine participants, you get a faster rise, 15 really high titers by day 15. So the infection takes 45 days and the vaccination 15. They get an increase after the second vaccination. And at the time of delivery, the antibodies to S1 and RBD were higher in vaccinated women, but the IgGs for S2 and N were higher in infected women, women who had been infected, for example. Fetal IgG for S2 and N were lower in the cohort of vaccinated women, while fetal IgG for S1 and RBD didn't differ from those of PCR-positive women. So these IgG antibodies are found also in the fetus in response to infection and in response to vaccination. And they also say, which is interesting, fetal IgM was negligible except for those 4 cases, consistent with no evidence for direct exposure to vaccine-derived antigens.
Right. That was going to be my question, actually.
So there's slight differences in the transfer ratio, right? But in the end, the bottom line here is that either from infection or vaccination, the mother develops a good antibody response. The kinetics are slightly different, and it's transferred to the fetus. I mean, what we don't know is, how protective it is, but it's certainly they're there, right?
So do we know that when we get injected with the vaccine in our arm, that it all stays within muscle tissue where the depot of vaccine was delivered, or does it get distributed through other parts of the body?
So a lot of it stays in muscle tissue, and the rest of it is picked up by local phagocytic cells like dendritic cells or monocytes that go to, but they only will really go to the local lymph node.
So that there's no circulating vaccine?
No.
So there's no, but what if you injected it intravenously into an animal that was pregnant? Would it get into the fetuses?
I don't know, but I would guess that the placental barrier would stop it.
Well, it doesn't stop IgG.
You're talking about the nanoparticles, right?
Yeah.
Right.
Well,
But
Yeah.
the
but the
IgG
IgG
There are
has
cells that
something-
go across.
The IgG has a
cell barrier.
specific receptor.
yeah, the
Yeah.
IgG has something specific to transport it.
Yeah.
But don't cells cross the placental barrier also?
Mm-mm.
No macrophages or
I don't believe so. No.
okay, all right. Just a query.
So they say this is the first time it's been shown that the antibodies are transferred from the mother to the child after infection, and they also say that the vaccine elicits a rapid rise and effective transfer exceeding the transfer rate observed in women with a third trimester SARS-CoV-2 infection.
Mm-hmm.
And they say that the titers in the neonates are substantial and potentially protective. So I think the point of this is that after vaccination, you clearly get transfer of antibodies to a variety of viral proteins, including spike, into the fetuses, and they may be protected as a consequence, right? And of course, as we said, you get, actually, they say here, Brian, that spike-specific IgG and IgA antibodies are in maternal breast milk, potentially building another line of defense. So,
I usually only think about IgA as being the one that is transferred.
yeah.
I am less familiar with that, but Steph can correct us.
All right. Well, let Steph correct us because I have no horse in this race. By the way, Dixon, when I got my mRNA vaccine, I dreamed of mRNA for a few weeks. So
Across the blood-brain barrier.
Who got into it? I'm just kidding, folks. It doesn't do that. It's cleared rather rapidly within a couple of, in the animal studies.
It's remarkable. It's
cleared
actually
very
remarkable.
rapidly.
Yes, it is.
It is remarkable.
In fact, there's a relationship between how strong the immune response is and how quickly the vaccine is cleared, which kind of makes sense.
Right. So the axillary lymph nodes are the ones that are doing most of the work.
I believe so, yes.
Yeah, okay.
That means here, Dixon.
Yeah, it does. It does, actually.
Axillary is a nice way to say armpit. Right,
That's
armpit
Richard
is not as nice.
axillary lymph nodes work even
So
better.
I thought this was an important study until we have vaccines for the young ones. The ones, they'll be protected if the moms are vaccinated. So
That's
moms should be vaccinated. Yeah,
But only for
And
the
this is-
first 6 months after birth. Then the
Yeah.
half-life
Right. Then they have to do it on their
body
own.
starts to fail.
This is great news, and it's also not especially surprising. I mean, given that there's a mechanism that the placenta has evolved to transfer IgG to the fetus, gives you an idea of how important this is to the survival of mammals in general.
Oh, yeah.
This is our general strategy for protecting infants and newborns, is that they get antibodies from mom, and then, breast milk provides more until the infant immune system is able to mature and, become exposed to all the things that infants immediately expose themselves to by putting everything into their mouths.
it's a consequence of having a complicated immune system. Now in insects where it's RNA based, the babies are ready to go.
Right.
Adaptive immunity. Why do you have to take so long?
I tell you.
So the key here is that you should be vaccinated before the third trimester because the transfer is better in the second trimester.
You should be vaccinated as soon as you can get
As
vaccinated.
soon as you can get it. In fact, But if you're pregnant now and you're not, you should be vaccinated.
And,
right?
and if you're pregnant and in the first trimester and thinking, well, gee, should I get vaccinated? The answer is yes, you should get vaccinated.
That's right.
Because the earlier you do it, the better.
Okay. Now we move. So that was our snippet. We had a nano snippet, a snippet, and now we have a paper.
Right.
Well, they're all papers, of course, but this one,
this is a whale of a paper.
This is
It's
a whale
big
of
one.
paper. And Dixon, I thought you would like this, Dixon. There's a lot of animals in it.
Well, I'm going to actually push this up on my screen now so that we can all follow along.
Yes.
It's published in PLOS Pathogens. It's a research article. Molecular fossils illuminate the evolution of retroviruses following a macroevolutionary transition from land to water by Xiaolu Zhang, Xuanhua Huang, Shen Gong, and Guangzhu Han. They are from the College of Life Sciences in Nanjing Normal University, which is for teachers, right?
Yes. I think we've talked about Nanjing Normal University specifically. There was some other normal university.
Well, normal. That's when
Nanjing?
you told us that it was for
yes,
teachers.
That's a teacher's college.
And that was the first time. And then I remembered it.
Yes. Good job.
In Nanjing, China. And this is very cool because I didn't know a lot of this stuff. Now, I didn't know that the ancestors of, all right, so cetaceans, whales, dolphins, porpoises, right? Cetaceans their ancestors went from lands to the oceans over 50 million years ago. I didn't know that. That's all. What did they do? Stand on the beach and say, hey?
No.
Right. So I was actually, I didn't remember the exact date. And I was, when I read this, I thought, that's really evolutionarily not that long ago.
No, it isn't. It isn't.
You know?
Yeah. So one of the things you can do when you go to a natural history museum, Vincent, because we point this out to all of our students whenever we talk about this, in terms of evolution, is if you look at sort of the large whale skeletons that you can see in different places, you can actually find hip bones.
Yes.
Yeah.
Because their ancestors were all land-based. And so they still have hip bones, even though they don't have those back legs.
Yeah.
And in fact, there are there are at least a couple of fossils of transitional forms, Ambulocetus, which is, it's a whale that still has some remaining legs, and Ambulocetus walking whale. And as they point out in this paper, whales are very closely related to hippopotamuses.
Which I didn't know. Right. I had no idea.
Which, if you think about it, kind of makes sense too, because hippopotamuses are halfway to being aquatic.
Yeah, they're pretty much always in water.
They're always
in
water.
So they have a common ancestor. Hippos and whales have a common ancestor, right?
That's right.
Oh yeah, because they say the earliest known cetaceans, pachycetids, are aquatic waders.
Yes.
So they start out by wading around and they like it.
Dip their little toes in
It's working out well. They're getting plenty of food. And so the toes become less and less necessary and become fins.
Well, and especially even in the hippo case, the other problem is that their skin is so thin that they actually can't be in the sun.
Right.
And so they go underwater as sort of a sunscreen.
Right.
The interesting thing is that, I mean, here was an open ocean. It had no major large predators. Imagine that as a niche to fill.
Yeah.
But there used to be. There were predators in the dinosaur age. So those huge.
Of course.
Well, and there were fish. Well, there were still large predators in the form of fish.
That's true.
But there were in the coastal areas, there was enough access to these niches that they could go into.
See, this is the stuff that the science programs like to make TV programs out of, right?
Of course.
And it is interesting, but you got to also do the hard stuff, which is what this paper is too.
So what's a molecular fossil, Vincent?
Hold on a sec. It's a good question. I just want to finish this evolution because to me it's just it's new stuff. So
you can never finish evolution.
The ancestors of whales, they walked on land, so they transitioned to water, and they lost the legs, right, and body hair, dermal glands, and they got other things. They got streamlined. They became filter feeders, etc. And now there are two main groups of whales. There's the mysticetes, the baleen whales, and the odontocetes, the toothed whales. That's going to be important.
Yes.
And as it says here, the hippos have a common ancestor. So the question here is, today cetaceans are infected with lots of viruses, and they list them all. There's your typical list: adenoviruses, astrocercoviruses, coronaviruses, enteroviruses, herpes, influenza. It goes on and on. You name it. But we have no idea, when they got these viruses, where they got them from. But to answer Dixon's question, we can do that with retroviruses because they convert their RNA genome to DNA. It integrates into host cell DNA. And if that host cell happens to be a germ cell, then the DNA can be passed on. As long as the host lives, of course, after the infection, the DNA can be passed on to the offspring.
I got it. Okay.
And now you can look, as we sequence more and more genomes, we can see, and they're called endogenous retroviruses. That's what a molecular fossil is, because you can see, and these endogenous retroviruses, they can go in the genome, and then they sustain mutations as the genome mutates, and some of them are inactivated, so they no longer make viruses.
Right.
And then, as we'll see, you can do molecular clock calculations by comparing the sequences of these, and that's what they do in this paper. So as I say, they're a unique resource to explore these retros during this transition from land to sea.
And because retroviruses have been around so long and have been so prolific, pretty much everything has these ERVs, these endogenous retroviruses, and you can go and look in their genome sequence. We're full of them. So they're human endogenous retroviruses, HERVs. The porcine endogenous retroviruses have been a big area of research because people want to do xenotransplantation with pig organs. The concern is, what if the PERVs go into the humans? Are they going to, cause problems? And I guess these would be WERVs, the whale endogenous retroviruses.
That's right.
Yeah,
That's right.
And the important question that they're trying to get at here is, where did those WERVs come from?
Yes.
Did the ancestor have them and bring them into the water when they went from land to water? Or is it a situation where these organisms went into the water and then acquired retroviruses from the new neighbors that they were in contact with in the water?
And
so it's important also to think about Vincent's observation that the hippo-like animal was the progenitor for these large swimming organisms that now feed on a totally different source of food than their ancestor. Their ancestor was probably a strict herbivore. And these whales are not strict herbivores at all. They feed on, the filter feeders feed on mostly krill, right? Mm-hmm. So that's sort of hunting in a sense. And all the toothed whales, how remarkable to go down 3,000 feet and seek your prey out using sonar or these little bleeps that's remarkable. That's absolutely, in 50 million years, I'm sorry, that's just incredible.
You go from grass to calamari in your diet. Yeah.
Exactly. Big calamari. We're talking about.
So Dixon,
do not order that at the restaurant.
The sperm whales are the toothed whales, right?
Yes.
Yeah.
Are there
not
others?
The only ones.
There are others too.
You've got beluga or toothed whales.
Killer whales, dolphins.
Killer whales, definitely.
Oh yeah, killers. So the sperm whales, they chase the giant squids, right?
They do.
Yeah.
They eat them.
Sperm whales actually remarkable. It's frightening to think of what their world is actually like. It's no visibility whatsoever at that level, and they're seeking out a gigantic mollusk.
They seem to be okay with
gastropod.
They do. Yeah, they do great at it. And in fact, that extended square head
that's right.
of the sperm whale that's full of what used to be the most valuable oil in the world the whalers were all going after.
Spermiaceae,
that's probably a buoyancy organ partly. Yeah, there you go.
I have a paperweight on my desk.
It's a whale.
Which is a sperm whale. I got this.
Doesn't it magnify that clicking noise also?
It may also magnify the clicking noise, which one theory is that's how the whale ship Essex got sunk. They were stuck in a doldrum and they were doing some repair work, some carpentry on the ship and hammering.
Oh, you're kidding me.
And that's when they got hit.
Oh, the whale charged them and hit them? Yeah.
Yeah. So the whale ship Essex was the inspiration for Moby Dick. It's one of the great sea stories. There've been multiple books written about it. I think Nathaniel Philbrick had one.
You picked it. You picked one of those books.
Yeah, I think that was one of my picks. It's a really good read.
Wow.
Yeah.
They got stove in by a whale.
Now, I mean, I find blue whales amazing because they're so big,
yes,
right?
They're huge.
Absolutely.
And they are baleen. Those are baleen whales, right?
The old whalers back in the wooden ship days did not go after blue whales because they sank.
Right.
So they went after sperm whales were another floater. And they're found in the Pacific, and they would sail all the way from Nantucket, because that's the center of all this, around Cape Horn up into the Pacific, and they'd hunt for a couple of years in the Pacific after the sperm whales, which float because of all the oil in their heads.
Right.
Many years ago, we took the kids to Vancouver Island, and we went out on one of these carved boats.
Yes,
to see the whales. And they have a lot of kelp floating on the surface, right? They're big tubes, and they're floating. And they're covered with krill, and we could see the whales. They would come up and tilt and lick along the side of the kelp. It was so cool. Very cool.
Goodness.
All right. Anyway, back to viruses.
Yes.
Endogenous retroviruses, and they go into the germline, the genome, and they can make infectious viruses, which would then infect other cells. They can also move around by a process called retrotransposition. The DNA can be transcribed to make an RNA. The RNA is reverse transcribed to DNA. It can go integrate somewhere else. And this has happened over years, and it amplifies the numbers of endogenous retroviruses. So 8% of the human genome is ERVs at the moment.
And in fact, the extent to which this has happened is one of the indicators that you can get in the molecular timing of this event. So that's why they serve well as fossils.
So if the retrovirus DNA goes in and it's quiet, reaches a dead end, you don't see a lot of copies. But if it's reproducing and transpositioning, then you'll see a lot of copies. So they took advantage of all the genomics that we have, and a lot of cetacean genomes have been sequenced. Not all of them, 25 cetacean genomes. And they found 8,724 ERVs in these.
I don't think there are any laboratory wet lab experiments in here.
This is computational biology.
Yeah, this is a computational. It's a lot of computational biology and it's heavy duty stuff. So I'm not detracting from it by saying that. But it's the nice thing about going after these ERVs is once you've got the genomes, then you can do these types of analyses and construct all of this.
Yeah, you can determine what happened millions of years ago, which is really remarkable because these are timers, these ERVs. Yeah. They're timers.
Yeah, and we can't do lab studies from 50 million years ago, unfortunately.
But these are obtained using today's cetaceans, right? You get their genomes and you can see, that's why it's a fossil in their genome, what happened many years ago. So
and by the way, you do not need to harpoon the whale to collect this, in case anybody's thinking this is a justification for the so-called scientific whaling that some places do. This can be a totally non-invasive sampling.
In fact, I like to show my virology class a movie of a drone flying over a whale and capturing whale breath. In a petri dish, and then they can see breath. They could look at the virome of whale breath. They have a lot of different viruses in whale breath. So they find ERVs in 6 mysticetes and 19 odontocetes, okay, and 8,724 ERVs. And, they say this is consistent with the ubiquity of ERVs in vertebrates. Pretty much all of them have them. And so then they say, okay, let's take a look at these and see if we can classify them further. So they have 315 distinct lineages. And the cetacean, each of these forms a monophyletic group, which means that each one is probably one or more independent invasion events. So 315 at least independent invasion events of the retroviruses gave rise to these 315 different ERVs. So then they, as Brianne pointed out, they have these 2 hypotheses. We have for the origin of these. They infected the land animals, right? That's the land to water scenario, the LTW. And then there's the secondary host switching, where they're already in the water and they get it from some other non-cetacean animal. And so they can begin to parse this out by looking at the sequences of these ERVs.
And this is an open access paper. And if you look at figure 1 when you're not driving, then you can see a very, very nice graphic illustration of this, where they show the 2 hypotheses, and then they show the tree, the classification tree of the extant cetaceans, and how this would distribute in the case of those hypotheses. So they're really outlining what they're testing here.
And here in the land to water image, they have a hippo there.
Yes.
On the left.
So the evolution of whales, sorry to diverge back to that, but it can't have a single animal origin, can it? When you've got 2 different kinds of whales? Yeah, it looks like you had at least 2 different events.
They split after going to the water, is what the available evidence seems to say.
Is that true?
I believe that's what is.
So this molecular thing should go back to a single unit before it gets back to the hippo-like animal.
Yes. Right. So you have your common ancestor there. They and the hippos are on land or very close to land for a while. And the hippo diverges off, and then one branch of that goes into the water, subsequently diverges. They gave the date for the subsequent divergence of the toothed and baleen, I think, didn't they?
Yeah, I think so.
So before that, they all had the same common herbs. That's correct.
Yes. So that's the basis of this, that it was a subsequent, right. So the 50 million year ago event is when they go into the water. Mysticetes and Odontocetes have been estimated to diverge from each other in the late Eocene, 36 million years ago.
Okay. Wow.
So something went into the water, and then it split into these two lineages after that. Mm-hmm. And so if it had the virus when it was on land, then you would expect the outcome that they outline in Figure 1, that the both of these groups of whales would have the same very, very closely related herb. Right. Because it would have been inherited from their common ancestor. Right. Whereas if it was picked up after they were in the water, and after that split, then you've got another 36 million years of evolution during which a baleen whale could have acquired a retrovirus, and it would only be seen in the baleen whales.
Exactly.
So, in fact, of these 315 lineages, right, 298 of them are found in both Mysticetes and Odontocetes.
Yes.
Right, which is consistent that it infected the common land ancestor, right?
Right.
And they find in general the copy numbers are low, which they say suggests that once they got in the water, they stopped being active for whatever reason. Huh. Maybe, who knows? There could be a lot of reasons for that.
Buoyancy.
Right. And some of them are closely related to the hippo herbs. And in fact, some of them are even in the same place in the genome as the hippo herbs. So that really shows that they were in a common ancestor, right? That's
which
really
suggests that it infected maybe the common ancestor of hippos and whales way back before either of them got to this.
Which begs the question, how long have hippos been in existence?
I don't know. Don't know. And so that was for some of those, but then there are other lineages that clustered with herbs from even-toed ungulates other than hippos.
Other than hippos,
right. So some of the hippo insertions are the same place, which is really quite amazing. So 298 herb lineages arose through endogenizations before the last common ancestor of modern cetaceans, and they got pretty quiet in the genome. There are not a lot of copies. Now, 17 of the 315 lineages are specific to sublineages, right? Some of them have them and some of them don't. So that's more consistent with getting it after you go in. And they find them related to a variety of mammals, including, Chiroptera bats, right?
Really?
Sigmodon. What is that? What is that? Is a rat or something? Or
it's I used to know that.
Sigmodon hispidis. It is a cotton rat. Cotton rat. Yes. Which are used for adenovirus pathogenesis study, and a whole bunch of other mammals. You could see the pictures of them in the paper here and get a kind of a clue of what they are. Yep. Even, bovidae, cows even. And you may ask, what the heck? How did a cetacean get it from a land animal? Well, they actually say that in the discussion, they say, that may sound weird, but actually killer whales have been seen feeding on land animals.
Yes.
So it could happen.
They will eat anything that goes into the water that looks the right size.
Yeah. So I
don't know about baleen whales. That might be a little harder to get to.
But the baleen could get it. They could get it from another whale that got it from a killer whale. Yeah. Who knows? So 17 lineages infected cetaceans in the ocean after the land to water transition. That's very cool. So both theories are correct. The land to water and the host.
The land to water is more common apparently for these sources of the earth.
And that kind of makes sense because if you imagine these large mammals going into the water, there probably aren't a whole lot of things that are obviously closely related to them. And so the viruses of the water organisms are probably not going to be tropic for these mammals that are suddenly going into the water.
Yeah.
What about the seals and the sea elephants and those other
mammals,
non-cetaceans that also are largely aquatic? I presume this group is working on them now too.
I don't know. Maybe they're into whales, cetaceans. I mean, maybe another group is. That's a good question. They must have their own nerves as well.
Yes.
Exactly.
In fact, Dixon, I was thinking the same thing and have been sitting here Googling to try to understand the phylogenetic relationships there. So I don't have an answer for you, but I've been Googling that exact same question.
I would actually expect this same group to probably go. I can't imagine that they are specialist whale bioinformaticists.
Whale herbs.
They must have had different origins on land animals, though.
Right. Yeah. That was what I was wondering.
Sure,
yeah. So you could do the same type of analysis for any of those. You could do seal herbs, and see where what is their common ancestors, probably something bear-like. I don't know.
That's what it looks like.
Yeah. So that, go back into that and see if they have bear herbs or if they have distinct aquatic acquired herbs.
What's interesting is that, complex life began in the ocean, and they went fishes and they crawled up in the sea, right?
Exactly.
And so
then some of them decided that was a bad idea.
Yeah, so apparently
wait a minute.
But the retroviruses are thought to have originated originally in the oceans.
Right.
And we did a paper a long time ago on TWiV estimating the origin of retroviruses about 450 million years ago, which is just before they were land animals. So they arose in the oceans probably, and then they crawled onto land with their hosts, and then they went back with some of them. Yeah, it's so cool. Another thing that you can do, which is very interesting, I want to just explain briefly to listeners. So at either side of the retroviral DNA, once it's made into a DNA and it integrates, there are two repeated sequences called long terminal repeats, LTRs. And they're both the same when the provirus integrates, when the viral DNA integrates and becomes a provirus, okay? But then once they're in the genome, they start to diverge because you have independent, they mutate according to the genome, and they're mutated independently because during reverse transcription, they are regenerated from the genome, so they're identical. So they say you can calculate the genetic distance between the two LTRs and get an idea of how old. Isn't that cool?
Yeah.
Just brilliant. Just brilliant. Wow. And so they do that, and they say it basically is congruent with what we think happened, right? That most of the herbs invaded genomes before they went into the ocean, if you calculate the age of these things. It's very cool. And they also looked at the SHS lineages, and they say, unlike the LTW herbs, these SHS seem to have proliferated more recently. They're newer. So they say the genetic difference between the LTRs peaked for all these 14 SHS lineages after the divergence of mysticetes and odontocetes. And they say, they could even be actively moving around today. And they could even be infecting whales. And they make the point of saying that we don't know the pathogenic consequences of these. Maybe we should look into it because, whales are kind of endangered and we don't want them to die off. So it's an interesting, they're currently proliferating and infecting whales. Very cool.
Whales are actually making a comeback, Vincent. So there are lots of humpbacks out there right now that there never used to be.
Yeah.
So that's a good sign, actually.
Do any nations continue to whale, Dixon?
Yeah, Norway and Japan. Those two.
Japan is the only nation, that whales out in international waters.
Correct.
Norway only whales in their own waters.
And why are they doing that? For dog food.
Seriously?
Dog food.
They're doing it for the reason we spend billions of dollars giving subsidies to farmers. They're doing it because there's a political reason to subsidize the activity, and so it's being done for bad reasons.
It's very sad.
There is also indigenous whaling.
But I was just thinking.
Yeah, in the Pacific Northwest of Canada, I think. And I don't know if this is any indigenous tribes in Alaska.
Oh yeah, I think there is.
But some of the Inuit or other tribes up in that area have special permission to hunt whales. That's a very different activity from what Japan or Norway does, because they are restricted to following their traditions, which means they like paddle out in wooden canoes and somebody stands there with it's very it's not going to have a significant dent on the whale population because they don't get very many of them that way.
So so Japan whales all around the world is what you're saying?
Japan whales all through international waters. Yeah.
And they have these huge ships,
massive factory ships and they go after the sinking whales, like the blue whales and such, the baleen whales, where they'll shoot a harpoon into it. And they've got this huge ship. They can. I think the harpoon actually shoots air into the whale so that it floats, and then they just haul the entire whale onto the ship and cut it up. And then, as Dixon said, they have it as dog food, or I think you can actually buy little packages of whale blubber if you're so inclined. But
it's a sad story, actually.
It's bad for such magnificent.
One of my good friends at
animals.
Rockefeller was Roger Payne. You might not know that name, but he became famous for recording humpback whale songs. And he gave a talk at Rockefeller while I was there, which was touted as the most well-attended talk ever. People knew what he was doing, and then he was like a maestro. You could see the notes, and the notes took up the entire width and breadth of the screen, because they were like 20 octaves above and then 20 octaves below, and he at that point he didn't know what they were for, but it turns out that these are male whales that are trying to out-sing the other male whales to get them to go away so they can have their way with the female whales.
Well, there's also a whole set of other calls and there's local communication and long-range communication, and because they're social animals.
Tell Vincent how
the mothers are speaking to the offspring in the pod and all this. And in fact, that, in addition to active whaling, is one of the other major risks that whales are having these days. And it may explain some of the beachings.
That's right.
Because the noise pollution that we have in our oceans, which is massive, everything from ships' engines to Navy sonar, incredible amounts of noise, and the whales can't hear each other.
But they can hear each other over thousands.
They can, but not if somebody's running active sonar pings.
Absolutely remarkable. Absolutely
so they get lost and they get beached. Is that the idea?
That's one theory behind beachings, and I don't know if that applies to all of them, but it's one of the things that might be going on.
A couple of other points I wanted to bring up. First, they say cetaceans have more interaction with terrestrial and semi-aquatic animals than intuitively thought, because I thought, how's a whale going to... For example, killer whales have been seen feeding on terrestrial animals and seals. So they could get viruses.
Seals for sure.
Yeah.
Little baby seals.
And what was the other? They say this may only be a small fraction of all the ERVs, because we haven't done that many genomes, right? There could be more. And it would be interesting to look at that. And they say, here's the other thing I wanted to mention. They say there's no evidence that these ERVs that infected the cetaceans after they went in the ocean came from fishes. Even though cetaceans have been feeding on fishes for a long time, that don't seem to have gotten ERVs from them, which is kind of interesting, right?
We don't get fish retroviruses either.
We don't.
It's probably just too different.
Yeah, I suppose.
Too much evolutionary distance.
So that's it. It's so cool. So two different ways that retroviruses, cetaceans acquired them, either before they went into land or into water or after. And you can use these sequences to calculate that. And they say, this is great for retroviruses. Other viruses is more of a problem. They say, there are lots of other viruses, but it's hard to know their origins, because we don't sample them all that much.
Right.
But I was thinking that there are, integrated fragments of DNA for a variety of viruses in all genomes, and we did a paper recently about that, right, on TWiV, where they look at these fragments of accidentally integrated, not ERVs in it, not needed for reproduction, but just pieces of genomes from a different virus. And you can do clock analysis and so forth. So I'm sure they could do that as well for these other viruses, adenoviruses, reoviruses, all kinds of viruses where a piece of DNA may be made and accidentally integrated, and it just stays there because it has no effect, right?
Yeah, they're harder to find and harder to analyze than ERVs. ERVs are, well characterized and they stick around in the genome and they got the LTRs and all that. But if some fragment of an adenovirus goes in, well, you've got to search the whole genome for anything that looks like any fragment of any adenovirus that's ever existed. So I remember discussing this when that paper was on the show. Was it's tricky to do those analyses and then know when that went in, but that is exactly the type of thing that you could try to do. And what you'd really want for that would be more cetacean genomes, I guess, which is going to be tricky to get.
Yeah, because your number of those integrations is probably going to be low, and so you need to look at a whole lot of genomes.
All right, there you go, folks. Something new, something different, I should say. I think it's just cool. All right, let's do some email. Hey, Dixon, you remember email?
Yep.
Can you take
Want me to do one?
Take that first one, Dixon.
I'd love to. Stephen writes, "Hi, Twivers. I'm sure by now you've received a number of alternate virus names based on the suggestion of Professor Henze at Northeastern. Here are a few more. Measles can be Weaselbergs. Mumps can be Trumps. And rubella can be Rubibella. That would make MMR virus vaccine, the WTR vaccine, which I wish I could have made the R and the F, but I'm not that clever. In any case, I was happy to be vaccinated against MMR when I was a kid, and I'll be very happy to be vaccinated against WTR as well. Keep up the good work. I know Daniel starts his segments with some quotes. I'll leave you with a favorite of mine from Louis Pasteur. Quote, "Science knows no country because knowledge belongs to humanity and is the torch which illuminates the world. Best, Steve."
So Dixon, last time,
I missed
Jamie
that.
Henze sent in, you said, you were talking about not naming viruses after a country. Why don't we name them after people who've been bad?
People who've been bad. Oh,
Right.
I see. That adds relevance to what I just
lean
read.
into the negative connotation of having a virus named after you.
I was thinking a little bit about this after that episode, and I remembered the story of Lord Jeffrey Amherst and realized that smallpox could be Amherst virus.
There you go.
Gotcha.
By the way, this quote is great. Knowledge belongs to humanity. I wish more people would respect that. It means
I wish humanity would take possession of it.
Yes.
Exactly right.
Not all of humanity is. Brianne, can you take the next one?
Sure. Elsa Maj writes, "Dear TWiV team, I am writing from Turku, Finland, where we are having a very exceptional summer with highs around 30 degrees Celsius. I discovered your podcasts in the spring of 2020 and have become addicted. Thank you for your scientific, but nevertheless humble approach to various topics. I am grateful for getting facts, but I also enjoy the way you discuss things. Since my training is in biochemistry, now retired, I do not understand everything, but I still learn a lot. I am also happy to know that some of our COVID-19 experts listen to TWiV. An absolute additional plus for Vincent was his praise of a manual shift car. I once in a while drive a car with an automatic shift, and I feel like meeting an old friend when I get back into my car. But I also have a question comment. I listened to NPR Politics July 12th podcast discussing the COVID-19 situation. They used the expression transmitting much more rapidly and spreading very rapidly. Is that okay? But then they went on to worry about the Delta variant becoming the main strain, with about 50% of cases in the US being Delta. Then adding that that in low vaccination areas, the Delta constitutes about 80% of cases. What I found alarming was that their concern seemed to be the Delta variant rather than the low vaccination rate. Also, they commented that there is alarming news from Israel that the Pfizer vaccine does not prevent disease. The addition that it does prevent serious disease came later. Of course, I did not understand why a vaccination passport is toxic in the US. Do you care to comment? You seem to have a real uphill struggle, but please going. With greetings, Elsmaj, who says that they don't expect to know how to pronounce it. Oh, Elsmai. The Maj is pronounced as Mai. And I apologize for all of the ways that I mispronounced it.
I don't think that transmitting more rapidly is correct, Elsmai. As we've mentioned before, it's more fit. It's displacing other variants. So I don't think it's okay, but many people say that, and
that's become the common phrase.
Yeah.
It's the more transmissible variant, and it's it could be, but we don't have any evidence saying that. And I think it probably isn't. I think it's probably got some partial immune evasion or something like that. But, yeah.
So you're right. We do have an uphill struggle, but it's all right. This and that's why unvaccinated. It's the dominant variant. So the unvaccinated people are what are getting it. If it were not dominant, some other variant would be infecting them, right?
Right.
Quite straightforward. Yeah. So the report from Israel, which we talked about briefly sometime before, it doesn't prevent infection. They said but we don't care if it doesn't prevent infection. It's disease, and the vaccine does prevent moderate to serious disease.
Right.
Right.
Yeah. Yeah, and this is really, vaccines always end up being victims of their own success. And it's happened on a much shorter time scale with the COVID-19 vaccines. If you remember back when these were being developed, and I know that was, 10,000 years ago in 2020, the whole discussion was about preventing disease. Yeah. And we on TWiV, we tried to kind of beat this into people's minds that,
that's right.
When we get the trial results, they're going to be about preventing disease. We're not going to know about preventing. We talked about sterilizing immunity. We don't know that we shouldn't expect the vaccine to provide sterilizing immunity. And then, especially with the mRNA vaccines, it turns out they do provide it's really robust. The infection rates, if you just actually look at infection and not disease, are really, really low among vaccinated people. So we got more than we expected.
That's
right. We got these amazing vaccines that do protect against infection most of the time. And then what's being called breakthrough cases are people getting infected back when the vaccines were being approved, these would not have been called breakthrough cases. These would have been called successes because the overwhelming majority of people who even get these breakthrough cases, which is like already we're down to a small minority of the vaccinated population, end up getting very mild or, asymptomatic disease.
Yeah.
And they don't end up in the hospital. And there are a very, very small number of people who do end up in the hospital after being fully vaccinated, and there have been a small number of deaths that can happen because nothing's 100%. But my gosh, if you look at the numbers on this, your odds of having a bad outcome from SARS-CoV-2 went from, wow, that's pretty scary, to, okay, I drive, I do many, many more risky activities than this. I shouldn't freak out over this.
So the population of the world now is 7.7 billion people, and we've vaccinated 3 billion of them.
Wow.
I just checked with the Johns Hopkins
website. That's a lot, but it's still not enough, right?
It's still not enough, but that's more than I expected.
Yeah.
It's half.
Yeah. It's unfortunately, though, very unevenly distributed. And so there are many places where it's less than 1% being vaccinated.
And
the vaccines differ.
So what do you say about this vaccination passport?
Right.
Don't forget that one.
Is it really toxic here?
Yes.
Well, it is in Florida, where all the cruise ships are.
I'm not totally sure if the question is, why is it toxic politically, or why is it just a bad idea?
Political. It's political.
I think in terms of why it's a bad idea, there are a whole lot of issues with access and how we would set that up. And I don't know that a vaccination passport is really necessarily what we want. But politically, it's just not.
All the cruise ships are pushing back on this one.
It depends on how you. So I've said since this first came up when early in the pandemic and people were talking about immunity passports, which were an absolutely awful idea because there was some proposal that if you've had the virus and cleared it, then you would get one of these passports. And I blogged about this. I tweeted about that. No, you do not want to do anything that encourages people to catch this virus because it's just
correct.
That's absolutely the wrong way to go.
That is right.
And then the vaccine passport, of course, came from many of the same pundits. Oh, now we're going to have this social gating mechanism because apparently some people are really into writing about social gating mechanisms, and it's got a lot of the same issues. As Brianne pointed out, very important issue is access. And early on in the vaccination campaign, a lot of people wanted the vaccine, couldn't get it. Now in the U.S., we are much less in that situation. People who want a vaccine can probably get it. I'm sure there are parts of the country where that's not entirely true, but it is widely available. So then why don't you do a vaccine passport? Well, the question is, what do you use it for? And I am generally opposed to any kind of a gating that is going to restrict people from ordinary daily life. Right. So I'm fully vaccinated. I'm a huge fan of vaccines, and I do not want to show up at the grocery store and have somebody say papers, please. Right. Whether that's a vaccine passport or any other kind of I just no, that's just not where we want to be. And that's not how we do things.
What about going to another country and they require it?
Right. So if you're talking about specific situations, for example, if you're going to travel to Australia or New Zealand, someplace that managed to at least locally eradicate SARS-CoV-2 and has had a slower rollout for their vaccination because they excluded the virus, because they did all the right things early on, they're going to have strict quarantine measures, and they're going to say, okay, if you're they may have different measures. I don't know actually what the current situation is in Australia or New Zealand. But there may be countries that have a situation like that where they could say, all right, if you've been vaccinated, we would let you in on an accelerated schedule
Iceland,
or something.
Is that one of those countries? Right?
Okay. That, you're going with that country's rules because you're visiting them.
Correct.
It's entirely up to them.
But sporting events also have these rules now too.
I would not want to see the U.S. implement something like that because we can't even vaccinate all of Mississippi. So if we're going to tell people from other countries, you can't come here if you're not vaccinated, then we're going to have to kick out a lot of our own people.
Yeah, but it's not a right to go to finals at Wimbledon, for instance, or it's not a right to be able to go to a football game or a basketball final. Those organizations have a right to say, we only want people here who are not going to aid in the spread of this disease.
Right. If you've got a very specific situation, right, like that, where you're forming a bubble, then I think it's totally legitimate to require vaccination.
Sure.
And I think it's completely legitimate in healthcare settings.
Right.
Or any other setting where people are going to have to be working in close proximity to mandate, all right, if you work here, you've got to get vaccinated.
Like schools also, right? I
am in favor of schools mandating it for everybody who qualifies, yes.
So the mask requirements are a better way to look at these things because everyone can wear a mask. Nobody objects strenuously to that. Right?
No, I don't think. I think some people do, Dixon.
Some people do, but that's something you can require more easily and not discriminate.
Exactly.
Yeah, New York City subway, you have to wear a mask. Everybody wears a mask.
If you walk into a place that requires a mask and you don't have one, they will give you one.
Or they'll point you out the door. Yeah,
I've been to many places like that already. Yeah. But it's very interesting to see that happen.
Back in 1960, we went to Europe, and in the passport was this yellow thing, which was a smallpox immunization proof, right?
Jeez, all right.
And maybe some others, and
yellow fever too.
It is probably not unusual, for nations to have vaccination requirements for travel.
Yeah,
that's right.
I agree. I don't want to walk around with papers requested, but I think
Yes.
for certain situations,
This is-
we
Yeah,
hear
so
it.
the vaccine passport idea I oppose because it implies that this would be a general purpose thing for access to daily life.
Yeah.
That I'm
Exactly.
against.
Exactly.
But if there's a specific argument for why everybody in a place needs to be vaccinated or should be vaccinated, like a school, like a hospital, like
Sure.
Wimbledon, then yeah, that's fine.
Yeah, and I can also imagine that there are some very small fraction of people for whom vaccination is contraindicated.
Yes.
Yeah.
And
of course.
it's one thing to have that conversation with your school to say, listen, this is why I'm not vaccinated. Let's have a conversation about
Show a
what I'm
doctor's
going to do.
note. Yep.
It's another thing to have to do that at the grocery store.
Yeah.
Indeed.
Have it delivered.
Alan, can you take the next one?
Yes. Simon writes, team with invincible values.
It's TWiV.
Yes.
Good.
As I fight the fight on social media, a thought struck me. People worried about vaccines, including, of course, anti-vaxxers, love pointing at adverse reactions to vaccines. Now, some of this is bogus and some of this is vaccine specific, aka allergic reactions to some random chemical used for good reason. Of course, only affecting a tiny minority of people. However, some low-level adverse vaccine reactions are things we also see as adverse virus reactions. As you know, there is evidence that some of the vaccines may cause a little clotting and some may cause a little myocarditis. And with the news this week, some may cause Guillain-Barre syndrome. Now, I know the numbers are low, only slightly above population normal levels, likely out in the 1 in 100,000 to 1 in 1 million level. If I understand the occurrence of these adverse reactions is higher as if I understand the occurrence of these adverse reactions is higher as reactions to the virus than to the vaccine. Yes, in some cases. So here's my question. In terms of a given individual, is there a link or trigger? In other words, if I took the vaccine and got Guillain-Barre syndrome, am I literally unlucky? Or does this indicate that I am a person with a higher risk of Guillain-Barre? And if I caught the actual virus, then the virus itself would have been even more likely to trigger that same adverse reaction. This seems to me to be an important question, important because of the biology, but also important because, in a sense, it means if a person gets vaccinated and is unlucky, then they really aren't unlucky, as odds are, if not vaccinated and then they caught the virus, they'd anyway probably have the exact same adverse reaction. So actually nothing really lost. And in fact, by being vaccinated, I reduced my chance of that adverse reaction. If this relationship is valid, then it changes the way to think about adverse reactions, as now it's not just that you did the right thing for everyone and we're unlucky randomly, but even at the personal level, it was the right thing. No idea if what I'm saying makes sense, but I just found it interesting how many adverse reactions are the same for vaccine and virus and more common in the latter than in the former. Simon, my understanding of this is that you are absolutely on the right track and that people have been looking at this for quite a while. And I'm not aware of really conclusive results on it, but the idea that there is, for example, a predisposition to Guillain-Barre syndrome, and that's, what caused it in a particular vaccination case. I think that's the rationale behind every vaccination form asking you if you've ever had Guillain-Barre syndrome. That this may be a contraindication to a vaccine. So there may be some personal predisposition to this. And I think others on the panel probably have more intelligent things to say about it.
So I think that the logic that Simon is using here makes a lot of sense. I think that it's not possible to do the conclusive experiment because we can't take a person who gets Guillain-Barre in response to vaccine, unvaccinate them, go back in time, infect them, and see if the same thing would have happened, which is really kind of how you'd have to do that. You can't do that. But if we look at sort of association type studies, it does seem like there are some predispositions here, but we're probably never going to be able to give an exact answer here.
Yeah, I think
you could get at proxy measures of this in animal studies, and I think some people have tried to, but the models are not perfect.
Yeah, I think for rare events you could do association studies, but for say a sore arm, more sore than somebody else, you're never going to figure that out. That's just not specific enough. But maybe GBS. But people are working on it for sure.
Yeah.
But that's very good logic.
That's a great thinking,
Really
and an excellent line of research. And as I say, there are people working on this for exactly these reasons.
All right. The last one is from Chris. I recently found out I could get HPV vaccine fully covered by insurance, human papillomavirus. I'd previously been too old. Would you get younger? Maybe it would be a good reminder to your listeners that this is available. I got the first 2 shots, but then I went on an extended work trip, and I'm 3 months overdue for the 3rd shot. I'll get it ASAP, but I can more fully empathize with people who don't make it back for subsequent doses when life gets in the way. Yeah, that happens for sure. I heard it's not effective against strains that I may have already been exposed to. Does that mean I'd continue to spread any strains I've been previously exposed to, or just that I can't clear them? Chris in Colorado. No, it doesn't mean that at all. It means that if you've been previously exposed, the vaccine is going to have less of a benefit for you. And so therefore, it is not recommended, right? That's it. If you've already been exposed, it's just that the vaccine doesn't have as much of an effect. That's it.
Yeah, and I think they recently they raised the age recommendation on HPV vaccine, and I forget. I'm certainly still not.
I have them up here. Here, let me read. So 11 to 12 is recommended. It's everyone through age 26.
26.
And anyone older than 26 not recommended. However, some adults 27 through 45 may decide to get it based on a discussion with their clinician if they did not get vaccinated when they were younger. Vaccination of people in this age range provides less benefit for several reasons, including that more people in this age have already been exposed. Okay, So you talked about it with your physician.
Vincent, what kind of vaccine is it? Attenuated or
It's a virus-like particle vaccine. It's the single capsid protein produced in either insect cells or yeast. It assembles into a virus-like particle. It's adjuvanted. And it is one of the human vaccines that prevents
cancer.
Prevents cancer. It also prevents infection, actually. It's Yes, so I never received it because I'm too old. And I didn't get younger, Chris, like you did.
Oh. Chris's name is Cassandra?
That's right.
I remember when my kids went for their HPV, my son said to the nurse, "Why do I have to get this?" And she said, "Ask your father." It's like, hey, I'm happy to do it, but not every father is going to be able to explain it, right?
Right.
That's true.
Oh, well.
I'm sure you over-explained it.
I don't even remember. It was a long time ago. He didn't actually ask me. He just said, okay, fine.
All right.
Okay. Oh, I forgot to ask my father that. I don't want to know.
Yeah, I don't want to ask him. All right, let's. Hey, Dixon, another part of TWiV is what we call picks of the week. Do you remember those?
Indeed. I remember. Listen, my memory is sharp as a tack right now.
I'm just kidding. I don't mean to insult you in any way.
You like to. I'm old, but I'm not that old.
Let's do some picks, Dixon. You can go first.
You got it. So what I thought would be more germane to the argument of a generalist discussion was just to show what's going on out west. In the United States Drought Monitor, the current map is telling the story that's been going on for 7 years in that same area. And if you don't think that this is predicated on the principle that we're undergoing a climate change that's going to be with us forever, basically, and it's just going to get worse. It isn't getting better. This is the worst drought they've ever had. This is making enemies from Colorado, who gets the water first, to all those other states that need that water in order for them to survive as a financial entity for agriculture. And the Colorado River just is overused, and now Colorado, of course, wants to expand its own agriculture. So it's diverting water from the Colorado River to various sources. So it's creating huge problems that are unsolvable using traditional agriculture, which is why I actually was an underlying reason for putting it in. it gives more reason for instituting things like indoor farming in cities and that sort of thing to reduce the pressure on outdoor farming. And this is a good way of just seeing it's a visual. No one doesn't understand this map.
This is good.
It's so absolutely simplistic that you cannot help but draw the right conclusions, which is, of course, the times they are a-changing. Who didn't know that? You know. And
you can we should point out you can zoom in on this.
You can
keep zooming in. So if you want, the West,
county.
It'll highlight the West, and then you go California, and it'll highlight California, and you want to say, oh, I'm going to pick Napa County, and you can get all the way down to see that there's exceptional drought in the wine district, and you can find out where your crops are, where your favorite food is grown, and a lot of the areas where most of our favorite foods are grown are going to be.
I hate to tell you, but Paradise, which rebuilt itself from a year and a half ago's fires, are now endangered again.
Yeah.
And the biggest fire in the West is now between Oregon and it's in the lower southeastern portion of Oregon. And there's another huge fire between Nevada and California that's just absolutely changing the landscape, and it's not going to come back right away.
Well, and meanwhile, here in the East, we're having the wettest July.
We are.
Which underscores your climate change point. Climate change is not everything gets evenly warmer. Everything gets more chaotic. So you get droughts, and you also get floods in different places, and it's
Dixon. So New York, southern New York, New Jersey, Pennsylvania, no drought, but one small area of Pennsylvania is abnormally dry. How does it get so focused like that, Dixon?
Well, that's a good question, actually. I'm not a meteorologist, so I can't actually give you the answer, but I'm sure it has something to do with geology and wind patterns and all kinds of other complicated things that we can't really predict very well. Wish we could, but we can't.
That doesn't have anything to do with politics, does it, Dixon?
I hope not.
Where's that? It looks like it's Schuylkill County.
Is that right?
Just outside of Philadelphia.
Yeah, I don't know why.
I don't understand.
It's very strange, isn't it?
I'm sorry, I don't understand.
But that's not like the Western exceptional drought. That's an abnormally dry. That's the D0 designation. So it's a little drier than the rest.
The West is a mess. Too bad.
The West is definitely a mess.
Yes.
Notice though, it stops at the Texas border. And Texas for years has had drought, and now they don't have a drought anymore. They've actually had flooding down there. So they've got an embarrassment of riches. They wish they could save all that water and sell it to the West, but all they've got is oil. Which is I'm afraid, boys, that we can't use that for our ag.
That'll just make things worse.
Yeah.
That would just make things worse. That's like throwing gasoline on the fire. Exactly right.
It's exactly like.
Yes.
Dear.
Brianne, what do you have for us?
So I have a comic strip. I was having a conversation. I was in a conversation on Twitter about the importance of T cells and antibodies. I know that's a big surprise. And someone tweeted this comic strip to me that they had drawn about the importance of immune responses. And it explains what antibodies are doing and what T cells are doing and sort of HLA and things like that. And I just think it explains things so well. And I was incredibly impressed with this. So, you've got the antibodies who are the police, and they might be fooled if the virus changes, it's outside, but the T cells are the detectives, and they are looking at something different. And so they won't be fooled. Each of us have slightly different detectives that can find different clues based on our HLAs. And so that's how it explains things in this very small comic strip, but it explains some pretty complicated immunology. I was incredibly impressed by this.
Rona might learn to trick the police, and now instead of stopping the virus, the antibodies are saying, yes, ma'am, the hotel you're looking for is right this way.
Yeah.
Nice. Very cool. Alan, what do you have for us?
I have a game that I just finished recently, so I do these fun picks occasionally. This is available on pretty much anything. You can play it on Mac, on PC, on Nintendo Switch, all the major gaming platforms. It's called Superliminal. And this is a puzzle game. If you like odd perspective art, MC Escher, weird games that take an unusual approach to the whole concept of a game, this checks all those boxes. So you're walking around. It's a first-person game, but you're not shooting things. You just walk in, and it's dreamlike. That's in fact the theme. Your character is at a sleep clinic, supposedly, and you wake up and you walk down the hallway and things are not right somehow. And you pick up a chess piece, and if you hold it out away from you, you can imagine the chess piece looks really big, right? Because it's obscuring a whole section of the room. And if you then let go of it, it is really big. You have this giant chess piece sitting there, which you can then pick up and break things with or do other things. And so all the puzzles in the game are based on changing perspective. You can watch a little trailer of it and see how this works. You pick up a model of a house and then make it much bigger, and then you can walk into it.
Cool.
You look out a window and then you can go into the scene that's there. It's all this sort of mind-bending stuff. Very fun.
Looks really cool.
It is very, very fun. There are a couple of puzzles at the end that I thought were kind of, that's not really in keeping, but the overall gameplay experience is excellent.
It's funny.
It's about I think it took me about 3 hours total. So it's not a big game, but definitely worthwhile entertainment.
I'm sure Dixon's going to do it once we finish here.
Right. He's a big gamer.
Oh, no, I was just doing it just now, actually. It looks like my real world, actually. I'm quite used to that now.
My pick is an article
what have you
from
got here?
northjersey.com. But birds are dying in New Jersey, apparently.
Jesus.
It's similar things happening in Washington, Virginia, Delaware, other mid-Atlantic states. And I noticed I saw 2 young birds dead on our lawn last week, quite young, in 2 different places, And so then my wife said, did you hear about this? I said, is it West Nile? And apparently not. They've ruled that out. It's the first thing I would look at. But they're saying if you find a dead bird, there's an email where you can tell them, and they may come and get it. I threw mine in the woods, which is what I always do, and then somebody will eat it. So, but I would just do a deep sequence and see what's in there on them. Right? They're saying, oh, it's very complicated to find out what's causing this. No, just sequence the damn birds.
I don't think so.
Well,
it could be a toxin though too.
It could be.
Yeah, I mean, you could get 20 different answers. I mean, they've been doing that with bees for a while for colony collapse, and everything is causing it.
So they say they have neurological signs, and they have eye swelling with discharge. Doesn't sound like a toxin.
During the West Nile outbreak, by the way, in 1999.
Yeah,
they actually put rat poison in the city parks to try to kill off the rats.
Yeah.
But they were so small in terms of the pellets, they looked like seeds. So the birds ingested them. And then they got diverted from making the diagnosis of West Nile by the fact that they found this rat poison in many birds in many city parks. So they had to, it delayed the actual discovery of West Nile by a little bit. I hope that's not what's going on here.
Yeah, I hope so. Well, you don't have any ideas, Dixon, do you?
None. No, and no more than you do. I don't think it's an infectious disease, though, because look where it's located. It looks like it spread from some central point and it went out this way. If you think that's an infectious disease, okay, but these are migratory birds for the most part, right? So this should be happening in a lot of other places right now because they've all fledged. Basically, all the songbirds have fledged by now.
Yeah. Yeah, it's around the Northeast, but it's not the whole Northeast. It's
selected states.
Mid-Atlantic into yeah. We don't have it around here, at least not that people have seen.
No.
We have 2 listener picks. We have one from our live stream the other night, 2 Listening 1. the New York City area, there's a living kidney donor transplant facilitator, and they do amazing work. It's called Renewal. So renewal.org, a comprehensive resource for kidney donors and recipients. So, yeah,
Cool.
That's great. I didn't know about that. So it's for potential recipients and donors. That's great. And M writes, hello, TWiV team. I wanted to share this beautiful Bacteriophage Wax Seal from Cognitive Surplus. As a virology nerd and a lover of stationery, I'm obsessed and very close to buying one. They also have a wax seal with DNA and a variety of science-related items, including clothing and notebooks. I promise I am not sponsored by this company, but they probably should be sponsoring me because I recommend them to so many people. There's also, and so this is very pretty. It's a nice bacteriophage. Yep. You can put it in your, if anyone seals envelopes with wax anymore.
And if you don't seal envelopes with wax, which may break in the mail anyway, but if you are just interested in this concept of cool science-related stuff, or you're shopping for gifts, then I was browsing around this Cognitive Surplus site. They've got a lot of really cool stuff.
They do.
Yeah, they do.
They do.
The great women of science, beer, pint mug, or if you prefer something a little different, the great beards of science. They've got
a core sample
different,
from the Earth.
but just lots and lots of different novelty items with scientific themes to them. It's very cool.
Very cool. So Emma's also wondering how many emails you get compared to how many you read. Do you read most of them, half of them, less? Less. Less than 10%, I would say, Em. Get a lot.
That's read on the show.
Read on the show.
Yeah.
Some of them you don't want me to read, actually, because, well, for various reasons, which you can probably figure out. And they get deleted very quickly.
Would this also percentage have changed since the pandemic?
Yeah, the number of emails went up hugely since the pandemic, for sure. But also, we never used to get nasty emails. I was always saying that, gosh, our listeners are great. They're so nice. And then none anymore. Most of them are nice. Most of them are nice, but there are some who
I suspect that a large proportion of the not nice ones are actually not listeners.
Probably.
Yeah, that's right.
Like, they're not people who are sitting through a 2-hour show of serious virology. They're people who are flying off the handle and heard that we discussed something and they think that we don't endorse their particular belief system. So
now, and also people that write emails and they have just making stuff up, right?
Yeah.
I get the common theme is I get this all the time. I used to love your show, but now you're into politics too much, so I'm not listening, which is just BS. We're not into politics too much, okay? So, and I tell them, you're dreaming, goodbye. I usually don't answer.
No, I don't answer
any of them, but once someone said, Dixon makes a lot of noise, you need to edit it out. I said, I don't have time to edit Dixon's noise. I have other job. I'm a professor. And you know what they wrote back? I'm sorry, maybe you get a paper cut, huh?
What?
Because I'm a professor and I shuffle papers, right? So that just shows you should never answer a nasty email because you get nastiness back. It was my mistake. Sorry. I couldn't resist.
My noise making is proof that I'm still alive.
Yes.
You're not noisy at all. You're fine. Okay. Thank you for your listener picks. That's TWiV 781. You can find the show notes at microbe.tv/TWiV. You can send us your questions and comments, twiv@microbe.tv. You send them, you never know when we're going to read it. And if you like what we do, consider supporting us, microbe.tv/contribute. Dixon Desbaumier is at Racaniello.org and livingriver.com. Thank you, Dixon.
It's good to be back.
Great to have you. I'm glad it all worked out. And looking forward to another 80 years.
It's very generous.
Brianne Barker's at Drew University. BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
Alan Dove is at alandove.com. Alan Dove on Twitter. Thank you, Alan.
Thanks. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology, and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.